Back there. Okay, wonderful.
Thanks.
Okay. I'll break a leg.
I will break my legs. Okay. Look at the cords back here. I probably will. Yeah. It's going to be interesting.
But you got it all connected, it looks like. So.
You do. Yeah. Shall we start? I'll start soon. They start a quarter after. Okay. Shall I sit? Yeah. Okay. That's fine. Not want my phone on me. Hey! Wow, man, you didn't have... Hello. Good luck. Thanks, man. That was awful nice of you. I was sure that you were going to be bronching. You're just gonna see things you've seen before. I would have been happy to give you a private show. You know, it's not what it sounds like. Maybe it is. I think it is off.
So, about 1, and that usually gives time for questions.
Okay. It should work. It should be right about it. If I'm going over, will you send me a signal?
Sure.
I've been known to do such things, and I can definitely wrap.
Okay. I'll stand on my head or something.
That's a signal.
We do. It's a great team to work with. It's really a pleasure. The new folks who come in, I think, are very much in that mode. So it's going to be fun. Just about since July. Yeah, he did all that. And he's doing quite well. Master's course as well. Oh, right. And Helena's doing, some of that sweet fellowship. What we need is
other things going on.
Yeah. But we, and Brian has really been great working with Terry. We really need next is getting other basic things. Oh, yeah.
Oh, but you need an basic researcher.
So, even if you're in, so understand. first year fellow this year. They just the first place that they cut money on the divisions that don't make money, right? But we have a second and third year fellow. Third year fellow is what he did is he's an MD patient that did his graduate work with David Goes all. No, so I suspect he's trying to hold on to it. Our second year fellow, on the other hand, is great. She may be on the road, and she's got a family in the West. So I will talk to her. She is wonderful. I don't know where to go. Try to point her out. We'll have a new fellow starting. We had to stop our fellowship for a couple of years. It is really one of the reasons to do this. The fellows are really great. The residents are great. Yeah. Yeah, they are. There's a commonality of purpose, right, and enthusiasm. It's just a commonality, right. Good afternoon. I'm Warren Regelman. I'm co-director of our Pediatric Pulmonary Division. Welcome, and congratulations on braving another sub-zero day. We have a very wonderful speaker today, Lucas Hoffman. He comes to us from balmy Seattle, where I'm told it's remarkably 20 degrees, and that's above zero. Although it is cloudy, and we have the sun today, so that's good. We have something to be thankful for, I guess. Luke did his training in chemistry at the University of California at Berkeley and received his PhD from UCSF as well as his MD from UCSF, and then went on to do microbiology, particularly molecular microbiology at Seattle. And he's had some very interesting insights into the microbiome, the bugs that inhabit our upper respiratory tracts, even gut now, and also some very interesting insights as to the microbiology of our cystic fibrosis patients' airways. He has numerous awards and support from NIH and from the Cystic Fibrosis Foundation, and I think you're going to very much enjoy hearing his talk today on what to expect when you're expectorating. Luke?
Thank you, everyone. Oh, is this a laser pointer here? You should have asked. Laser pointer? I did. I should never have picked this up. Okay, we're going to talk about hand washing today. Very important topic. Sorry. Why don't I quit?
I just have to switch inputs, I think.
Should I unplug and plug in again? It didn't. It doesn't screw in. In pulmonology, we say, smoke 'em if you got 'em. We don't really say that.
Yeah, maybe try unplugging it.
Okay. We might have to use one of your connectors. Do you have those connectors? Okay. Well, while we're waiting for the new connector, Thank you, Warren, very much. Very generous invitation and very generous introduction. I have to say that as I was putting this talk together and just thinking about it, I'm more than a little bit intimidated about talking to this department and this division about exactly this topic, because I'm going to be talking about chronic respiratory infections in children. And in particular, I'm going to talk the most about the paradigm, the most common of these chronic infections that we study, which is cystic fibrosis. You want me to take this out? And when I think about the University of Minnesota, and particularly the Department of Pediatrics, oh, that looks better. I think about a lot of people that have contributed tremendously to exactly this field, and on whose shoulders I sit, maybe stand. And the first person that I think often about, and that comes to mind, you can just pull that out if you want, is the great Warren Warwick, who I had the distinct honor of meeting just a few years ago, and who was instrumental in many ways in pushing forward the field of CF research, CF care, and many of the concepts that he came up with. That's perfect. Many of the concepts that he came up with and his treatment approaches are approaches that I use. What do I do with that? Okay, so I plug this in here. Oh, this is fancy. My keyboard can't be identified. You know what? Who needs a laser pointer? We're fine. Okay. So here's Warren Warwick. And I'm not showing here what I consider to be the most important or the most influential paper, at least for me. But I'm just showing the oldest paper that I could download as a PDF from PubMed here, showing you how long Warren was in the game. He was very inspirational. And, of course, when I think about this department, I think about him, as well as the person I referred to as Warren number 2, my generous host, and a microbiologist and pediatric pulmonologist who also I feel I'm sitting on his shoulders as well when I talk about the concepts I'm going to talk about. I'm showing one paper here that Warren published in 1990, and I've read cover to cover many times during my own fellowship and since, because it was really, it was a very provocative study, an incredibly innovative study design that's never going to be repeated, I'm sorry to say. But it was really the first paper and the first study that I know of that tried very hard to quantify the effect of infection and the effect of our antibiotics on children with these kinds of chronic diseases like cystic fibrosis. And here's another dear friend and colleague that's sitting in the audience, Terry Laguna, who, as you can see, is the senior author on a paper that is on exactly the topic I'm going to be talking about that just came out a few months ago. And I will talk about this paper in a little bit and show you how it's opened doors to completely new areas of inquiry that I think are very important in these topics. So with that in mind, these are just 3 examples of the kinds of diseases that I find particularly interesting as a pediatric pulmonologist and microbiologist. And when I say interesting, of course, that's code or shorthand for incredibly frustrating, because these are 3 examples of chronic diseases or chronic infections that just don't respond to antibiotics the way that we think they're supposed to based on everything that we've been taught using traditional concepts. Now, for example, here's one group of children. This is one patient population. These are actually 2 patients of mine from the west coast of Alaska. These are Yupik Eskimos. They are adorable, as you can see, and this population, which includes tens to hundreds of thousands of patients in a fairly large area, get chronic airways dilation, chronic inflammation, loss of elasticity in the airways called bronchiectasis, and concurrent lung infections for reasons that we don't yet understand. And there are several native populations throughout the world that get that kind of disease. So I'll talk about that in a bit. Children who have plastic in their airways for any kind of reason get chronic infections, again, that don't respond as well as we think they're supposed to. But the disease that I take care of the most, and that is perhaps among the most frustrating, is the genetic disease cystic fibrosis, which I'm sure you've heard about quite a bit on this campus. And I'm not showing the picture, the face of one of my patients, but rather here a CT slice, a CT scan through the lungs of this young woman showing these bronchiectatic airways and in that patient as well. So what I'd like to talk about in relation to these kinds of infections and many other chronic infections today are, why do the airways of these patients get infected in the first place? We've learned quite a bit, and actually we've known quite a bit for a long time about why these infections happen. And what can we learn from, or what can we use from what we've learned recently to understand the pathogenesis of other diseases? And what have we learned about the function of healthy lungs? There's some surprising new information about that. And finally, at the very end of the talk, I'm going to pretend to use this information to be able to go forward and tell you how we can better diagnose and treat these infections. Spoiler alert, I don't know. But I will show you, I hope to show you that there is a big door open that didn't used to be open to the way forward, I think. And I'll show you what I think is the next wave of infectious disease management for these chronic infections. So starting with how these airways become infected, again, I'll just talk about a couple of these diseases. This is a schematic of airways from somebody with cystic fibrosis down here. This is a schematic cutaway of a normal airway. CF, as you probably know, is a genetic disease. It's an autosomal recessive disease and the most common genetic disorder in Caucasians, and one of the most common genetic diseases in the world. It's caused by a mutation in a single gene, the cystic fibrosis transmembrane conductance regulator, or CFTR, which regulates the flow of ions and fluids across epithelial cell membranes in several organ systems. And so while that mutation impacts the function of several organs, it's really the lung disease that's responsible for most of the illness and early death in our patients. And to oversimplify a very complicated disease process, what happens in the lungs is that the airways of our patients become obstructed with vast amounts of very sticky, tenacious mucus that don't move normally. What happens in the normal airway is that our epithelial linings secrete a small amount of thin mucus that then serves to trap everything that we inhale, including microbes that come through the upper airway, through the oral cavity, from the environment, and everything else that we inhale, like smoke or pollen or toxins. And they deposit on that mucus, and then that mucus is then removed with everything in it by the usual mucociliary clearance mechanisms, cough clearance, normal breathing movement, and we either expectorate that or we swallow it. In the case of CF, the mucus doesn't move out as normally. It just sits there and stagnates, including all of the microbes that are in that mucus, all of the toxins that are there, and that induces inflammation and ultimately, again, dilation, scarring of the airways, loss of elasticity, and bronchiectasis, which makes matters worse. And so these patients have a problem with clearance of their infected mucus. It stagnates, and you get chronic infections that are very difficult to treat. And unfortunately, still at this point in history, respiratory failure is still the most common source or common cause of death in these patients. This is a very simple schematic of the collection of disorders which we call non-CF bronchiectasis. And there are a lot of parallels with CF, and as such, we think about pathogenesis very similarly, and we also treat these patients very similarly. But the problem here is a little different in that the bronchiectasis seems to come first, and the poor mucus clearance comes secondary to that bronchiectasis. You can get bronchiectasis from any kind of toxic inhalation, a really bad local infection or diffuse infection. You can get it from genetic problems such as immunodeficiencies. And in our native populations, such as our native Alaskans, as I talked about, the Aborigines in Australia, the Maori, we don't yet know why they get these diseases. But their airways seem to become dilated first. And then with that loss of elasticity and the scarring, you still get mucus stasis and poor clearance as a result. And so you end up with the same problem. Infected mucus that stagnates, stays, leads to progressive respiratory disease and respiratory failure. So this is a very simplified model of how we think these chronic infections happen in the case of poor airway clearance, which is really the case in all three of the types of kids I told you about. Everybody inhales bacteria, microbes, from the environment through the upper airway, and then something interferes with clearance, which then allows chronic infection to be established and subsequent airway inflammation and lung disease with associated symptoms. And so because of the importance of microbes in this pathogenic mechanism, we use a lot of antibiotics in treating these patients to try to prevent the establishment of chronic infection and then to try to suppress symptoms once that infection is established. And unfortunately, while antibiotics are very useful in controlling symptoms and maybe slowing down the establishment of chronic infection, they're at best moderately effective at this, and they certainly don't halt the progression of disease. And that characteristic, the not quite adequate or unsatisfactory response to therapies that we think are supposed to work well, are what define these kinds of infections as chronic infections. They join a big list of infections that we refer to with this title, with their defining characteristic being they don't respond to therapy that we think is supposed to work as well as we think it should. And you get progressive symptoms and progressive lung disease as a result. And this is despite using antibiotics that our usual in vitro tests tell us should be very effective. And so when I say that, I'm being a little overly provocative purposely. It's worth reviewing why we consider this to be adequate treatment. And I'll give you a little history, which I'll bet you know, and be a little preachy. But reviewing where these concepts came from, the usual in vitro tests that we use to diagnose and treat and study these kinds of infections really come from concepts that were handed down by these two guys and many others who were not as well recognized. A German named Robert Koch, a Frenchman named Louis Pasteur, who together gave us a series of laboratory tools and microbiological concepts which we still use today, almost 100 years later, and which were remarkably effective at allowing medical science to tackle and understand and choose good treatments for a number of really terrifying diseases, some of the scariest, really rapidly fatal infectious diseases that mankind was facing at that point and still faces, such as sepsis or meningitis or pneumonia, each of which generally involve the infection by one species of microbe growing very rapidly, usually in a sterile liquid. And so the concept that they gave us, which were directed towards those kinds of infections, generally involved taught us to isolate the one microbe that caused those infections and then grow it in the laboratory, isolate it from everything else, grow it usually in a rich aerobic suspended laboratory culture or in an animal model, and then use those conditions to study these microbes, find out what makes them tick, find out what kills them, and then use that information to try to treat our patients. And as I mentioned, these methods are very effective against these rapidly fatal, usually one pathogen infections. But what we're left with, as I referred to, are a group of infections that don't seem to behave the way that those methods tell us they should, which we call chronic infections. When, again, their defining characteristic is that they don't go away, they can't be eradicated necessarily, but they just sort of smolder along. They also don't rapidly kill people, but they just continue despite our best efforts with these antibiotics that these methods tell us should work. And as you might be able to think of a lot of kinds of infections that fit these criteria, but if you scale up over human history and over the entire world, of course, these kinds of infections are responsible for a tremendous amount of suffering and illness and early death in people. And as with all of the problems that we don't do a good job at treating, they're disproportionately suffered by those of us who can least afford them. Now, I've shown you three kinds of chronic respiratory infections. There are, of course, many others. And these pictures I just chose to show you that different kinds of chronic infections in other organs, in case you hadn't thought of them yourselves. Chronic infections can afflict almost every part of the body, particularly parts of the body that are usually colonized with bacteria. And essentially everything that we put on or in patients can also become chronically infected. That is, they don't necessarily eradicate, or at least quickly, with usual antibiotics. So coming back to these three kinds of respiratory infections, it's important to keep in mind during this talk that I'm going to talk mostly about these, but they the details that I give you have implications for all kinds of chronic infections. And of course, I'm going to focus mostly on cystic fibrosis, because this is the best studied of all of these chronic infections, and as such it serves as a paradigm for all kinds of them. And so everything that I tell you about CF or bronchiectasis has implications for everything else, and I'll try to point that out. So with that in mind, this is the way that we generally, as you know, study, diagnose, and choose treatments for these kinds of infections, like CF. We take the secretions from our patients. We then culture them, swab them onto a selective laboratory medium, or multiple media. And when I say selective, I mean we've chosen those media to be able to grow the bacteria or microbes that we already think are important. We then further select out just a few visually distinct isolates, or sometimes just one isolate that we think is representative of all of these traditional pathogens. And then we further culture those isolates of a few species onto laboratory media again. Here, this entire plate is swabbed with one isolate. And we're testing susceptibilities to multiple antibiotics. These are antibiotic-impregnated strips here. And you can see the zone of clearance showing you how susceptible it is. So with these in vitro susceptibility tests of a few isolates, of just a few species, we then choose our antibiotics and we treat our patients. I'm leading you a little bit here. And this is the view that we get of cystic fibrosis chronic infections from these diagnostic tools, again, based on traditional concepts. I'll call these traditional methods from here on in. This is a slide that's put out by the Cystic Fibrosis Foundation every year, and you may have seen it. These are most recent data I've got. And they're from 2011, and they show you the proportion of respiratory cultures from all the CF patients in the U.S. reported to the foundation that were positive for each of these 6 traditional pathogen species and some subtypes of them. And they're broken down by age. So a couple of points to see are that far and away, children are most often culture positive for this organism, Staphylococcus aureus, which is a gram-positive bacterium. And then in adulthood, you more often see Pseudomonas aeruginosa, a gram-negative bacterium. So these are the 2 bacteria that you culture most often using traditional methods from CF patients. And using these methods in other kinds of chronic infections, you get fairly similar views. You can culture out a lot of the same traditional pathogens that you see in people with cystic fibrosis, including Staph aureus, there's that, Pseudomonas aeruginosa, Haemophilus influenzae was also on that other graph. So these traditional methods then give us a list of pathogens or organisms that we culture most often from people with different diseases. And this is the paradigm, or this is what we understood using traditional methods with regards to healthy airways. You can find, this is just one older paper that I found this phrase in, but you can find it all through the literature and even in textbooks. It says, recent bacteriologic studies have shown that the bronchial secretions of people are usually sterile in the absence of significant disease. So this is what we knew at this point, using traditional methods. So that's our view of these infections, and this is how well these methods work clinically. Many studies have looked and have not been able to establish any relationship between our in vitro susceptibility tests using these methods and how well our patients do. They don't predict who's going to respond or not respond to antibiotics. Many studies have also looked and not been able to establish any relationship between the abundance of traditional pathogens in our patients' specimens and how they do. That is, how sick they are. And even more frustrating to those of us who take care of kids, which I think is everybody in the room, or many people in the room, I'm sorry, Brian, a good fraction of kids who have very significant structural lung disease, inflammation, and symptoms, sometimes symptoms bad enough to need hospital care and IV antibiotics, have their cultures negative for any of these traditional pathogens, which makes it very hard to know how to treat them. So clearly, we'd like to do better. And there are many reasons why these methods may be inadequate in showing us, in choosing therapy and showing us what's making our patients sick. One possibility, as I alluded to, is that in all of these diseases I talked about, the respiratory diseases, there's a failure of clearance of toxic pro-inflammatory stuff. So infection is only part of a complex pathogenetic mechanism here. You've also got obstruction with your mucus, your nasty mucus, and inflammation, all of which are related to each other. Now, so we keep this in mind as we move on, but of course I'm a microbiologist, so we're going to talk about infection for the next 40 minutes, 20 minutes. So the next possibility I'll talk about today is that our traditional methods, where we just select out a few isolates of these traditional pathogens and then study their behavior in a rich suspended liquid culture, maybe that doesn't really represent how diverse these microbes are, these traditional pathogens, or how they behave inside an infected tissue. So with regards to behavior, there's now pretty good evidence that our suspended liquid cultures, single species cultures, are not very reflective of what's going on in these infections. And one example, there's good evidence from several kinds of infections that many of the bacteria in chronic infections don't infect suspended free-swimming or free-floating cells, but rather they grow adherent to each other such as these aggregates in an electron micrograph of CF sputum, or adherent to a surface, for example, on plastic. And this growth phenotype is known as biofilm formation, and biofilms tend to be very resistant to antibiotics. We also know that in these infected tissues, while we tend to grow our liquid cultures with rich media, very, very aerobic, with plenty of oxygen, that nutrients and oxygen can be really limiting into these conditions. And here's an example from a very nice study from over a decade ago where somebody finally looked and measured the oxygen tensions in the airways of people with CF and found that they're quite low. And under these conditions, you expect antibiotics not to work very well. So, allowing for this, what do traditional culture methods tell us about what these traditional pathogens look like over time? And what we've found, what is known and has been known for a long time, is that these methods tell us that these traditional pathogens change. They adapt to the host or something else over time. And that those changes are very diverse. They're not just simply adapting to the presence of antibiotics. We're not just seeing antibiotic resistance, but we're seeing changes in all kinds of behaviors that we wouldn't otherwise expect. And this is just an example from a study that we're wrapping up where we took isolates, 2,700 isolates of Pseudomonas from children, several hundred children, with CF over many years. And on the y-axis, each one of these lines represents a patient. Each dot represents a culture. This is age on the x-axis. And so each culture is then categorized into whether it represents the earliest phases of infection, that's initial. The blue dots are intermittent, that is, they have alternately positive and negative cultures. And the green dots generally represent persistently positive cultures, so that's chronic infection. We then took all of these isolates and ran them through a battery of very simple, high-throughput in vitro behavioral tests, including antibiotic susceptibilities, but lots of other bacterial behaviors. And what we found, as had been shown previously, but this is a big comparative study, is that there are lots of behaviors that seem to change over time in a fairly stereotypical way that distinguish early from intermittent from chronic infections, such as colors and the production of various exoproducts like proteases and in motility, like swimming, for example. So the view that you get then is that during chronic infections, if you just look at a few isolates, that bacteria adapt and change in a fairly linear and stereotypical way. But there are now many studies which show us that this view is quite limited. And here's the most dramatic, and I think the first example of exactly this. This is a group from London that took single sputum samples from individual patients and then isolated 50 or 100 isolates of Pseudomonas that otherwise looked the same on a plate. So more representatives than the single isolate that we would usually use of each morphotype. And in this case, they've tested the susceptibilities of 50 isolates from just one sputum sample to all of these antibiotics. And the susceptibilities are on the y-axis. And the point is that if you take multiple versions of the same similar isolate and test their susceptibilities or look at other behaviors, they're actually all over the map. That even though they look the same on a plate, they have very different antibiotic susceptibilities. So the better view is not of linear adaptation, but diversification. During chronic infections, these microbes seem to really change, and they go all over the place. They change their behaviors in many ways that our traditional methods are not representing. And in fact, if you believe the traditional methods, you can start to see some very surprising phenotypes. One example from our study was this one. Here is an isolate where we tested susceptibilities of this isolate to 4 different antibiotics from one point in infection. This is early in infection. That patient then proceeded to get all 4 of those antibiotics at various times for the next 8 months. And here's the only isolate we got 8 months later. And you can see the zone sizes, which show you that because they're bigger, it means this isolate was hypersusceptible to all the antibiotics this patient got over that 8 months. So it would be very confusing if you didn't consider diversification. It suggests that this patient's bacteria didn't even keep its susceptibility despite pretty heavy antibiotic therapy. And so this kind of diversification could help us to explain why there isn't a good relationship between our in vitro results and how well our patients are doing. So we now know that microbes that these traditional methods really underestimate the diversity of these microbes in lots of chronic infections. They change, they diversify over time, and we think those changes are driven by antibiotics, nutrients in the environment, and Brian Williams is teaching us a lot about what the nutritional characteristics of these isolates are, and oxygen, and also host factors as well as the presence of other bacteria. They're probably adapting to all kinds of things, which drives this phenotypic change over time. Oh, and Ryan, I put your name up there as well, is teaching us a lot about how bacteria interact with each other. And so our current limited culture techniques of just a few isolates don't provide an accurate view of that diversity. In general, we presume that one isolate represents everything that's there of the same species. And a better view that we should have of these chronic infections is one of communities of functionally diverse bacteria that are probably interacting with each other in a way that we don't model, and we're inadequately sampling this diversity. So this next study that I'm going to tell you about, I chose to try to drive this point home. This is a story about a subtype of Staph aureus called small colony variants, Staphylococcus aureus. And as I mentioned, Staph aureus is a gram-positive bacterium. It's the most common organism that we culture from kids with lots of different respiratory diseases. It's also a normal commensal, and it causes lots of difficult kinds of infections. And we also know that it changes over time, but those changes are relatively poorly studied in all of these chronic diseases. So again, on this graph, I just put this here to remind you that Staph is far and away the most common organism cultured from kids with CF. It's actually the most common organism cultured from everybody with CF now. And the foundation tracks a couple of different subtypes of various bacteria. Here they track methicillin-resistant Staph or oxacillin-resistant Staph. But one subtype they don't track are called small colony variants. And this is a culture of a small colony variant of Staph aureus from one of our patients next to an isogenic normal colony isolate from before in the same patient. And so you can see they're called small colony variants because they form small colonies, at least on laboratory media. Now, they form small colonies in vitro because they have metabolic defects. They grow slowly on our laboratory media. And because of that, because it takes either special media or longer incubations, very few clinical labs in the world detect small colony variants or isolate them. There are only a few investigational labs that do so. And what people have found is that if you look in chronic infections, you find them. The reason that they grow slowly is because they generally have mutations or are defective for producing specific portions of their key metabolic pathways. And in this case, this is a mutant in the production of thymidine. You can see this isolate, which was swabbed on this plate, only grows around this disc of thymidine. So they're metabolic mutants. And those same mutations don't just make them slow growing in vitro, but they also make them highly resistant to a lot of the antibiotics that we use clinically. So these are bacteria that can infect people that we don't detect that are very resistant to the antibiotics we use. And my laboratory got interested in this topic a number of years ago because of a surprising in vitro observation we made. We were just trying to build very simple binary two-species models of Pseudomonas aeruginosa and Staph aureus, the two bacteria that we culture from many kinds of diseases most often. And we would grow them together just in simple liquid cultures and then plate out that binary mixture onto blood agar plates at various times. So this is a culture after a couple of days together. And so on these blood agar plates, Pseudomonas, it's hemolytic, so it looks gray. Here are a couple of wild-type Staph aureus colonies. But the majority of Staph under these conditions grow as these stable small colony variants. If you streak them out onto a new plate, they're stable. They're mutants. They keep their phenotype. And this doesn't happen with Staph alone. It's due to an interspecies interaction. So we went on to show that in the presence of either Pseudomonas or specific antibiotics, both of which are found very often in our patients, such as in their airways, most of the Staph will eventually grow as these undetected, slow-growing small colony variants, which are highly resistant to a lot of the antibiotics we use. So the implication is that in the context of these infections, Staph may often be growing or always be growing in a form that we don't detect and that resists our therapy. So this suggests that perhaps Staph is around more often than we're even detecting and may be more important in the pathogenesis of these diseases than we thought. So we decided to test that with a very simple, short study, single-center study, by collecting all of the Staph we could, including all of the small colony variants we know how to detect, from 100 kids at our hospital over 2 years, and then correlated those small colony variants with clinical outcomes. And we found small colony variants using these methods in about a quarter of our kids over that 2-year period, which is about the same prevalence as MRSA and persistent Pseudomonas. But the most concerning finding came when we correlated those small colony variants with various clinical outcomes. And this is lung function here. This is the lung function value, forced expiratory volume in 1 second, which is a measure of airflow through the airways and is the most common lung function measure that we use in following the disease in most of our children and adults. And what we found is that in the kids that had small colony variants, they had lower lung function at the beginning of the study than did the patients who did not. This is percent predicted. And they lost even more lung function during that 2 years. They lost 8% of their lung function during that FEV1 during 2 years, as compared with 1.8% in the kids that didn't, so about 4-fold more. So it raised the question of whether small colony variants were just more common in kids that were already sicker, or whether these are significant pathogens, or whether they're markers for something else that's really making them sick. And so to test that, we made a few, actually quite a few, a lot of logistical regression models, very simple models, where we looked at the relationship between small colony variants and loss of lung function during the study, controlling for various confounders, such as age or lung function at the beginning of the study. And in this model, there was this still very significant association between small colony variants and loss of lung function. We then started controlling for various other organisms, such as Pseudomonas here, which, as you can see in this model, didn't weaken that association, but also in this model where you're concurrently controlling for both small colony variants and Pseudomonas, Pseudomonas was not associated with loss of lung function during the study, nor was MRSA. And in fact, we couldn't find any confounder that we could think of that small colony variants seemed to be a marker for. So again, this suggests that either these guys, which we're not detecting, are significant pathogens in our patients, or they're still markers of worse disease in a way we haven't figured out. And another point that we found is that when we use quantitative PCR in the sputum of our patients with and without small colony variants, there's no evidence here that small colony variants, even though they have a growth defect in vitro, they seem to be able to maintain very high densities in the lung just fine, or at least in sputum. So it's not hindering their ability to be very abundant in these patients. So what I've shown you is that our standard methods miss a lot of bacteria, or miss very prevalent bacteria that may be very important in the pathogenesis of these infections, in this and other chronic infections. And so we could be missing at least important pathogens or markers of worse disease. And so this diversification and adaptation I'm talking about in chronic infections renders our usual traditional methods, where we select out various bacteria, particularly individual isolates, inadequate in sampling the diversity of these infections. And here is just one more very quick something. I've got more in the talk, but just for this point, about Pseudomonas that drives this point home. This is just a little observation we made recently, where a scientist in my laboratory swabbed out sputum from one patient onto a Pseudomonas isolate, a medium that selects out Pseudomonas, and happened to put a bunch of discs of antibiotics on here. What he found is that, as the clinical laboratory found, they did not isolate any Pseudomonas using the usual method from this patient. So this patient was called Pseudomonas negative. But on this plate, you can see the Pseudomonas is growing around this one disc. This is and this strip of trimethoprim sulfamethoxazole. This is, as far as we know, the only example, the first example of a sulfa-dependent bacterium. This is a patient who got a ton of Bactrim of sulfamethoxazole. And so here's another example of how our selective culture methods can really miss a potentially very resistant organism just because of adaptation and diversification over time. So in summary, I've shown you that individual bacteria change, they diversify, they adapt over time. And some of those changes can make them difficult to detect, but also difficult to treat. And so that raises the question, if we're missing some of these traditional pathogens, what else are we missing? And I'm talking about other kinds of microbes beyond the ones that we generally culture. So that brings me to the last possibility I'll consider of why we're not doing a good job, and that's that we're misrepresenting all of the bacteria that are really infecting our patients. So this is exactly the question that was faced by environmental microbiology about 50 years ago. Many environmental microbiologists made an observation that concerned them and that they needed to work around, which is now called the Great Plate Count Anomaly, where they would take samples from the soil and from water and look at them under the microscope and concurrently culture those samples using the best methods they have. And they found that only 1% of the bacterial cells that they could see under the microscope were culturable using those methods. 99% were not. So they knew they were missing things, as I'm suggesting we might be. So they invented and refined a series of methods, we owe them a lot in medicine, called microbiome methods, which involved extracting DNA, particularly the microbial DNA from those samples, and then using various methods, profiling methods or sequencing methods, to identify and quantify the microbes in those samples. And using those methods, they identified a lot of uncultured and sometimes unculturable organisms in environmental samples, and they're still doing so. So that raised the question of whether this could be the case for humans. And several studies in the interim showed, yes, there are a lot of bacteria that we don't culture using our usual methods in and on even healthy tissues. And those were the findings that inspired the Human Microbiome Project, which was established about a decade ago. And because, with its goal being to identify and tabulate all of the microbes that we can detect on and in healthy people. And as you can see, because we know that the lower airways in humans, the healthy lower airways, are sterile, the lung is not on this map. Well, a very wise environmental microbiologist named Ken Bruce at King's College in London about the same time looked at the CF literature, because his lab is right next to the hospital, and he decided that this looked like an environmental microbiological problem, that these disconnects between clinical and in vitro findings seemed a lot like their Great Plate Count Anomaly problem. So he decided to apply what was then the state-of-the-art microbiome method, which was a gel electrophoretic profiling method called TRFLP, to 13 samples of sputum from 13 different adults with CF, and showed and then published that there were a lot of bacteria that we don't culture, and some that we do in these samples. Not just abundant traditional pathogens, but a whole bunch of other bacteria that we don't culture. And it took a little while for that concept to catch on, but since that bomb was dropped about a decade ago, there have been now dozens and dozens, and I'm only listing some of them, not all of them, of studies that have confirmed and extended and built upon these findings and made them more clinically relevant even, using better and better methods. And I'm highlighting in blue here that study that I'm going to tell you about with Dr. Laguna as the senior author. So this brings me to the next of 4 points. I will get through soon, I promise. What does this tell us about the pathogenesis of other diseases? We know that now that CF is polymicrobial. And using these methods in lots of other chronic infectious diseases, we now know that they have similar kinds of microbial diversity. Here are some studies. This is one study from adults with non-CF bronchiectasis, all grouped together. There are now 2 studies that show very similarly that they're also very, very diverse infections. And there is similar diversity found in other kinds of chronic lung diseases using these methods as well. But the big surprise came when people started to use healthy controls in their studies and found surprisingly high diversity in their healthy controls. And in fact, now there have been a couple of studies where people have just done very careful microbiome studies of healthy people, taking samples through bronchoalveolar lavage and protected brush-induced sputum, and shown that, sure enough, the lower airways are not sterile. They have detectable microbes at appreciable densities, decreasing as you go from proximal to distal, and that are very diverse, including lots of uncultured bacteria, and that are different from what you find in the oral cavity, for example. So this brings me to the last point I'm going to talk about, which is the misrepresenting the diversity of all the microbes that are present in these infections. But before I go on, I just wanted to define the term microbiome, which you probably heard many times. And that just refers to all of the microbes and their genetic material in one particular place. So it describes not just who's there, but how they behave. And I'll also use the term diversity, which is a measure of variety in the community. Do you want me to wrap up soon, or can I go another 5 or so? Okay. So this is a measure of the variety of all of the microbes, the taxa, the species that are in a place. Not just the number of species, but the relative abundances. And the microbiome methods generally involve extracting all the DNA from a sample and then using various methods to just focus on the microbial portions of that DNA, usually using probes or arrays, or more recently using sequencing methods. And so the information you get depends on what kind of method you use, and it makes it hard to compare studies done differently. And it generally involves, as I showed you in the early days, a very qualitative, non-quantitative community footprint that gives you a good idea of what's there and what's not, but it's not high resolution, and it's good for comparing samples from different patients. But more recently, with the use of sequencing methods, we get much higher resolution information about who's really there, often down to the species level, as well as the relative abundance of these bacteria. So it's higher resolution, better taxonomic resolution, and more quantitative. And so to summarize all of these dozens of studies, including hundreds of aggregate patients in cystic fibrosis at least, we now know that regardless of the sample type you look at of these secretions, and often even in respiratory tissue, lung tissue from people, you can find traditional pathogens using these methods, as well as similarly abundant other bacteria that we do often culture, but the clinical lab calls them oral flora, and also a lot of bacteria that we don't traditionally culture, often at very similar abundances, particularly in kids. And there's a lot of variability in these microbiomes between patients. So one patient tends to have his or her own specific respiratory microbiome that doesn't change a whole lot in the short term. And it's that kind of variability which makes it hard to combine these patients together into large community-based studies or into large aggregate studies to try to correlate microbiomes with respiratory outcomes. However, a couple of studies have shown us a lot. And this is my favorite example of a study that is correlated with clinical outcomes. This is a really nice paper that was put out by a group in Michigan last year, where they looked at a series of patients who were stable, that is, they had no loss of lung function over a decade, and then a series of patients who progressed over that decade. They had loss of lung function. And they looked at the microbiomes in the sputum samples over the decade. So each one of these dots represents one sputum sample, and you've got diversity on the y-axis. What they found is that the patients who progressed over that decade had loss of lung function, or I'm sorry, had no change in their sputum microbiome diversity over that decade. But the patients who did progress had loss of diversity. That is, they tended to lose species, and it got simpler and more dominated by one or two species over time. And they went on to show that there was a good association, a good correlation between that loss of diversity and the amount of antibiotics the patients got. So we're back to that chicken-egg problem. Did these patients get more antibiotics and have this change in diversity because they were sicker, or did this change in diversity make them sick in the first place? And so we don't really know. And since these findings have come out, there have now been parallel studies in all kinds of chronic respiratory diseases, including some that we traditionally think of as infectious, like bronchiectasis, chronic pneumonia, or in intubated patients, chronic obstructive pulmonary disease, and even some diseases that we don't usually think of as infectious, like asthma and bronchopulmonary dysplasia. So this is a field that's just opening up. Now here are the results from that study that I keep referring to with Terry Laguna as the senior author, that I think is opening up a really interesting direction for CF research and for all of us. What they did in this study at the end is they correlated the change in particular taxa of bacteria with antibiotic treatment with clinical response to that treatment. And what they found is that the change in a genus of anaerobes that we don't detect, called Prevotella, correlated the best with clinical change. And this suggests then that the antibiotics that we're using to target traditional pathogens may actually work for reasons we don't originally intend them to work for. And so again, I think that's opened a brand new door, and it's a very interesting concept that will show us the way forward to try to figure out when patients get better and how to improve that. So the final little study I want to tell you about just in a couple of minutes was a comparative study that we just finished up. And I've told you about two diseases, CF and bronchiectasis, non-CF bronchiectasis. We wanted to compare the microbiomes in sputum microbiomes in kids with these two diseases and with one other disease, a disease called protracted bacterial bronchitis, which you have almost certainly seen. This is a wet cough that persists for greater than a month that generally gets better with antibiotics. And it's recently been shown by several groups that there's a bacterial component to this disease. So what we wanted to do was compare the most common and most abundant microbes called the core microbiota, or core microbiomes, in the sputum samples or BAL samples from each one of these groups. And what we found was that when you looked at those cores, they were very, very similar. You've got similarity on the x-axis here, which measures, again, the abundance and identities of these taxa. So they're greater than 90% similar, suggesting these infections start similarly. But then when you look at the sputum microbiomes from adults with the two chronic versions of these diseases, CF and bronchiectasis, they were not only different from the pediatric versions, the cores were, but they were actually very different from each other. So the view here is that, or the implications are that all of these diseases begin similarly. The infections begin very similarly and then diverge over time in a disease-specific way. And so, again, we're back to the chicken-egg problem. The question really is, do these differences reflect differences in underlying disease? Do they drive the disease, or are they caused by the different therapies that we're using, the antibiotics that we're giving, or both? So in summary, I've shown you that the lower respiratory tract, even in health, has diverse microbes, many of which we don't detect using our traditional methods, and that are likely shaped by imbalances in delivery and clearance of just the usual suspects from the environment, the upper airway, and the oral cavity. And as a result, it's probably not surprising that diverse respiratory infections have very similar beginnings, even when symptoms have already begun, but then they diverge over time. And these changes could be driven by all sorts of features of the underlying infected tissue, including other bacteria and antibiotics. And so now we're at a point in history where we have to try to figure out what's causing what, what's making patients sick, and what's causing the change in diversity over time, and how those two things are related. So finally, I'm going to pretend to tell you how we do better. How do we use this information to better treat and diagnose these infections? And as I mentioned, I don't know. But I do feel like we've made a lot of headway. These methods have shown us that our current methods aren't working. These results show us that our current methods don't work. They're far from exhaustive. We're undersampling the diversity of microbes. And so if I were a betting man, I would say to improve our outcomes, the future is probably going to involve a combination of better culture methods, more inclusive methods, plus the molecular methods I'm talking about. And the question is what exactly that's going to look like. We also now know that even the healthy airway is not sterile, and that this finding could be really important for understanding the pathogenesis of all kinds of infectious and traditionally not infectious diseases. And we know that focusing on individual pathogens in all of these diseases is probably not the best conceptual approach, but rather we need to come up with a better conceptual model for all of these chronic diseases and all kinds of chronic infections that may consider the microbial diversity in all of them, and that may allow us to treat these diseases better. So the door's open. I just don't know what the outside looks like. Now, the goal in all of these studies is to try to figure out what's making our patients sick and how to make them better. And with that in mind, the final little thought experiment I'll leave you with that somebody suggested to me years ago when I was very skeptical about all of these findings. He suggested, think about the last century when we put together our list of traditional pathogens that we thought made people sick. How would that list be different if we had used microbiome methods over the last 100 years instead of culture-based methods? I think that's an interesting thing to consider. So with that, I'd really like to thank you and for your patience. I'm sorry I went over, as well as my laboratory in blue here and my clinical colleagues, John LaPuma in Michigan, who is wonderful, my funding, and of course, study subjects and the families that participated in all of these studies. I'll take questions if we have time. Gail.
So, we were just having a discussion in our pulmonary rounds about chronic cough and actually came upon this protracted bacterial bronchitis. And I think the question for me is, and you had that very nicely with your microbes, airway inflammation, and mucus, theoretically, everybody should get something, right? So is there a difference in the airway or the mucus clearance? I mean, obviously there's a lot of diversity in the microbes, but, where do those other factors come into the kids who are, obviously kids who have asthma or, CF or bronchiectasis already have airway damage, but what about those normal kids
Right.
who get these other things and their risk factors?
I don't know the answer, but there are a couple of really helpful observations. In protracted bacterial bronchitis, Miles Weinberger showed that it's much more common for that to happen in kids who have some other underlying clearance defect, most often tracheomalacia, for example, or bronchomalacia. So that, to me, suggests that this is another case where you got your normal microbes, and some lesion or some underlying problem causes you to fail to clear the normal bacteria. So there's this new imbalance. So if that's the case, tracheomalacia is not universal in the kids that get protracted bacterial bronchitis, but it's common. Maybe there is a genetic difference, I won't call it a defect, in immunity in some of these kids. Or, and I think this is very plausible, for both this bronchitis, acute bronchitis, and for pneumonia, maybe it's just a bad local lesion from a viral infection that causes a local failure of clearance that then, or it could be more diffuse, that then leads these usual bacteria that are always there to suddenly not be cleared the way that they're supposed to. I think that's a very plausible possibility. And so then the variability that you're talking about, whether a kid gets it or not, is going to depend either on something underlying or the severity of the inciting event that caused it. You could say the same for toxic inhalations. I mean, we generally treat chlorine inhalations, for example, with antibiotics, and we know why that is, because there's probably a secondary failure of clearance, either immune clearance or mechanical clearance. So that, I think it probably has to do with some lesion that just predisposes to poor clearance. That, to me, that feels the best in terms of mechanism. Does that answer?
Yeah, I mean, I think the interesting piece of this is to really also try to look at the whole concept of airway inflammation.
Right.
Where does that come in?
You're right, I didn't mention that.
And, sort of, is there anything we can do about it?
Yeah.
Outside of antibiotics, because, again, we're all told that we shouldn't use antibiotics because we're overusing antibiotics, but,
Yeah.
So there's the circular logic type thing.
I know, and you're right. You're absolutely right. I mean, if you consider a secondary infection that's really common, like influenza, which generally leads to, if it's going to get really bad, it's because of a secondary bacterial infection. It's probably the host response to the influenza that is the problem, probably not the virus itself. At least that's what a lot of people think. In which case, it's inflammation that was the problem. And I don't know what the best way to treat that is, because I don't know for a fact, particularly if it's neutrophilic, that steroids are going to make a big difference. I need an immunologist to talk to us about this here. So I don't know what the answer to that is. I don't know what the best way to approach that is. It's one of the reasons that you notice I put up that slide and then I moved right along to the bacteria, because I haven't a clue what the best way to address the host response to the inciting event is, or to the ongoing infection. Yes?
So do you foresee maybe a probiotic someday where you have to inhale a more diverse population
Yeah.
of bacteria again and get back to normal?
I actually think that's the idea of reseeding with diverse microbiomes is very interesting. And I think for the diseases we're talking about, we are extremely far from reaching ethical equipoise to allow that to happen. But I think that it's quite plausible. I just don't think in the next few years we're going to see anybody nebulizing bacteria of any type until it's seen in an animal model, for example. But I think it's a very interesting idea, because I do think that this diversity, for example, could be controlling the expansion and behavior of the guys that are really making people sick, for example. Yes. Oh, okay. Yeah, I think it's a very interesting idea. And in fact, two interesting observations. There is now known to be a relationship between what you find in the gut and what you find in the lung, in people with CF in particular. There's probably communication between those two niches. And we also know from a couple of small and not terribly well done, but still pretty definitive clinical trials, that probiotics, oral probiotics in kids with CF seem to decrease the rate of exacerbations and make their lung disease, at least by some measures, a little better. So there's a plausible mechanism here. It could be that either modifying the gut microbiomes, alter the microbiomes here, could act through mucosal inflammation in a way I don't understand at all, because I'm not an immunologist. But it's plausible, and I think it's not as far in the future as we think it's going to be. I just don't know what that's going to look like, and I'm afraid I'm not going to be the one who's going to be doing it. Maybe I'll inhale some Prevotella tomorrow, but not yet. All right. Well, thanks for sticking around. Thanks. Oh, I unplugged my computer.
I just hope before anybody goes nebulizing bacteria, we do it carefully.
I know.